Boggs is here. Bonner. Clay is here. Dawson. Ellis. Paula. Green. Harrison's here. Last name James is here. Esterson's here. King. Lewis is here. Let's see. Grace is here, or excuse me, online. Ward is online. Don't forget, if you guys are online, you need to send the words before 9 a.m. Wilcox. Wilder is here. Bree. Morrison just came in. King is online. Whew. So today, just so you guys know, a little announcement, I'm only doing online office hours. So I'm doing office hours, but I won't be in office. I'll strictly be online. Let's see, what are some reminders? Reminders, let's see. Friday will also only be online office hours. What else? This lab for this week is virtual. So there's a possibility if you get it done early and I get it graded early enough and you did a good enough job that you wouldn't even need to come to lab this week. So keep that in mind. What else? That's it for reminders. Don't forget to do independent work. I'll be putting in those grades soon. Like I made an announcement last week for that. That was a written announcement. So now I'm verbally reminding you, whatever your grade is for independent work, I'm going to go ahead and put it in the system, which will make your grade drop. But keep in mind, none of that. He brings the light with him. None of that's official, right? Your grade is not official until I turn in grades, right? So when you see your grade drop dramatically online, don't be worried. I just don't want you to be surprised at the end when I do put in your grades, your independent work grades. Because remember that it's not due until the last day of class. So that would be a huge surprise and shock to the system. So I'm going to go ahead and put it in the system. And that way you can see what you're up against, so to speak. You can see what your grade looks like without independent work. And then you can work on bringing it up, so to speak. Does that make sense? I don't want you guys to be stressed about it. This is not your official grade. Your grade's not official until I turn them in at the end of the semester. So I guess that's it for announcements. I think. What else? Oh yeah, Veterans Day. Just remember, I've said this before, but Veterans Day, November 11th, we will be having class online only. Of course, I'll send you guys a link for that. Probably the night prior. I guess that's it. I can't think of any other announcements and reminders. Let's see. Bonner is online now. Green is online. And with 1 minute left, let's do attendance one more time. Aki, not here. Boggs is here. Bonner's online. Clay is here. Dawson is here. Ellis is not here. Paula is not here. Green is online. Harrison is here. Henson is not here. James is here. Kesterson is here. King is online. Lewis is here. Linda Q is not here. Morrison is here. Grace is online. Ward is online. Wilcox is not here. Wilder is here. Bree Parks is not here. And Wilcox is here. Oh, that's a good reminder. Okay, so when you want to join online, you need to make sure that you're logged into your WVSU Google account. Right now, someone's trying to join. I'm going to let them in. But sometimes I might not get the notification, so you might be trying to join in online, and it won't let you because you're not logged into your WVSU Google account. And like I said, if I don't notice it, then I won't know to let you in. So make sure you're logged into that. Any questions about anything? All right, nice and easy. Get started. Where did we leave off? Let's see. We definitely finished talking about the cell cycle. We got all the way through G1, S phase, G2. We got all the way through the mitotic phase, including mitosis and cytokinesis, those two overlapping things. But then, where we left off was talking about sort of like the exceptions to the rule. There we go, and that's this. The exceptions to the rule, if you want to call it that. And you can see here's the cell cycle that you're used to, that we've already talked about. We have something called G naught, which is the little G with an O, as you can see. I guess, you know what, hold on. G will be the first word for attendance, or I guess letter, if you will. G. Anyway, some cells do not always follow the classic cell cycle that I taught you, right? They don't always go from G1 to S to G2 to the mitotic phase, right? There's exceptions. So in some cases, they are in what we call, again, the G naught phase. This is when the cell is not actively preparing to divide. It's an inactive stage. And it's basically, you should think of it as kind of like exiting the cell cycle is what the cell has done. That's the way your book describes it. I think that's a good description. So it's sort of like paused in the middle of the cell cycle. It just kind of leaves it, if you will, right? Sometimes it's temporarily triggered or is temporarily triggered by external signals. You can learn a little bit more about that once we talk about DNA, we talk about genes and how that works. But for now, that's all you need to know. And sometimes cells just remain there permanently. And that is a good independent topic if you want to look that up. First of all, which cells are more likely to be in the G naught phase, right? And what are some of those external triggers? You can look that up if you want. As far as the exam is concerned, however, this is very unimportant because there's probably not going to be any questions about this particular phase. It's just an exception to the rule. Your book talks about it, so I included it as well. There's also a variability in the cell cycle length. Again, like I was mentioning earlier, not only does it vary within an individual, like for example, your kidney cells are different than your brain cells, right? And those are different than your nerve cells, right? It's very different, even within you. But obviously, as you might imagine, it's also different from species to species. And that's, again, other stuff you can look up for if you want. So, using that as an example, when we look at humans, the frequency range, the frequency ranges from sometimes hours and sometimes the entire lifetime. And again, that I think is a decent independent work topic. Like which of your cells go through a cell cycle quickly within hours, and which of your cells stay in the cell cycle for your entire life. You can look that up if you want. So yeah, the timing of this is controlled by internal and external mechanisms. And again, you're going to learn a little bit more about that later when we talk about how genes work and how proteins work. But for now, that will suffice. So are there any questions about this slide? All right. So next thing we're talking about are these little checkpoints. And this goes back to a question that Kesterson asked last week. You remember what it was, Kesterson? It had something to do with how the cell cycle is controlled. I don't remember exactly how you worded it, but that's what we're about to learn. We're going to learn how the cell cycle is controlled. As you already know, the whole purpose of the cell cycle, right, when it's repeating or duplicating itself, right, the goal there is to get daughter cells that are exact duplicates of the parent cells, right? So they want to make a perfect copy. However, that does not always work. Sometimes we have things called mutations, and we'll talk more about mutations in a different chapter, but this is your first formal introduction to mutations. They can be inherited by all subsequent cells, which makes sense. So imagine, again, you have one normal cell, and then it goes through the cell cycle, goes through mitosis, and it duplicates itself. Now imagine in that process, there was a mutation, right? There was a mistake in duplicating that DNA. Well, since it usually works And if you have that mistake, then therefore every copy after that is also going to have that mistake. Does that make sense? It'd be like if you had a paper and you took it to the copy machine and you copied it, and that first copy was fine, and then you took that copy out and you accidentally smudged the ink or something, but then you started using that to make copies. Obviously, every copy after that is going to have that mistake, right? Because there's that smudge. You smudged the ink, and now you're making an exact copy of that smudged ink, so it's going to be smudged, right? Same thing here. If the cell goes through mitosis and has a mutation, then every copy after that is going to have that mutation as well. So, for this reasons, amongst other reasons actually, we have control mechanisms, right? We have these checkpoints that Kesterson was asking about, and I really wish I could remember the exact way you asked it last week, but it was a good question, and here we go. So these checkpoints, these mechanisms, they ensure that the cycle pauses until things are good to go, right? So the cell cycle, it's not just on auto drive, right? It's kind of like stop signals and go signals, because amongst other things, your cells are trying to avoid these mutations. And a little side note, as you're going to learn later when we talk about natural selection, mutations are actually really important. Without mutations, we wouldn't have natural selection, and without that, you just wouldn't be here. So those are important, and again, we'll talk about the later date. So these control mechanisms ensure the cycle pauses when it needs to. There's few of them, and you might need to know this for the exam. So if you were taking notes, you might just, I don't know, circle that. I'll go ahead and circle it for you. Like this, if anything, this might be what you need to know on this slide. These checkpoints are located at the end of G1. They're also located between the G2 and mitotic phase transition. So I guess another way you could say that is at the end of G2 or at the beginning of the mitotic phase, and during metaphase itself, right? So there's 3 checkpoints, and we're going to talk about all 3 of them separately. But are there any questions so far? All right, so let's talk about the first one, the G1 checkpoint. This checkpoint determines if the conditions are favorable for division to proceed, right? So remember, if you can envision the entire cell cycle, right? That's the entire cell cycle. So you have this part, which is interphase, and then this part, mitotic phase. And remember, interphase itself can be broken down into G1, S phase, and G2, right? So here we are. We're talking about the G1 checkpoint, which is right here at the end, right? Right before you leave G1 and go into S. This is also called the restriction point. I'm not going to ask you that. At this point, once it goes forward, once it goes past this G1 checkpoint, the cell is then irreversibly committed to going through the entire cell cycle and doing cell division, right? Once you leave G1, it's committed. And that makes sense, right? Because what comes after G1? S phase. And as you know, and as you need to know, S phase is when the chromosomes duplicate, right? So if you're leaving G1 and you're going into the S phase, obviously your cells are going, your chromosomes are going to duplicate. And then obviously, after that, the cell is going to be committed to splitting. So some of the things it looks for in this checkpoint, we want to make sure there's adequate reserves of material and energy, right? If you're going to split your cell, that requires energy, that requires material. That's why I said last week, I think it was, during the regular G1, right, the cell is also growing and it's getting bigger. And also, it checks for damage to the DNA. So again, that goes back to the whole mutation thing. There's an enzyme that we're going to learn about later. The enzyme that does the DNA duplication during S phase, it also checks the DNA. It also checks the chromosomes to make sure that there's no mutations. So if all these requirements are not met at the end of G1, this G1 checkpoint, then it just does not move forward. So any questions about the G1 checkpoint?
This mutation, is it embedded in the DNA? DNA's telling it to create this?
Good question. I'm glad you asked that, because technically we're talking about no mutation. This is not a mutation, right? This is avoiding mutations. This is saying, in a perfect world, when it works, it will not move forward if there is a mutation. It will fix the mutation or not move forward. But that out of the way, yes, the mutations are in the DNA, but there's going to be a different chapter on that when we talk about mutations deeper. So for now, that's all you need to know is that, yeah, the mutations are in the DNA. And to remind you, DNA, right? That's genes, right? Those are your genes, and genes, as a reminder, are instructions on how to build proteins. It's literally a language, as you're going to learn later. So what happens is some of the words get screwed up, and then because the words are screwed up, and those are instructions on how to build proteins, then sometimes those proteins aren't built, or sometimes they're built the wrong way because of the mutation. But again, mutations will be a discussion in a later chapter. So any other questions? That was a good one. Any questions about G1? Excuse me, the G1 checkpoint. Okay, let's talk about the G2 checkpoint.
Wait a minute.
This, yeah,
If, go back to G1 checkpoint, if it's determined that there's damage or something wrong with the cell,
uh-huh.
does it destroy the cell or?
Sometimes, yeah, and that's a thing called apoptosis, and I don't even teach that because it's deeper than we go. But yeah, sometimes it'll kill the cell, sometimes it'll just fix the damage. Or obviously sometimes this whole thing doesn't work, and there's a mutation, and it's not fixed, and things go forward, and again, that's how we get mutations that stick. And that's also how we get natural selection. Without that happening, without this mistake, without this thing going wrong, we wouldn't have mutations. Excuse me, we wouldn't have mutations. But we also wouldn't have us. Like, for example, and you don't need to know this because this is just a random example, and I think I've said this before, humans, well, first of all, why should humans eat citrus fruit? I'm sure you guys have heard this, right? You know you need to eat citrus. What is it in citrus that's good for you that you're supposed to be eating it for? What vitamin?
Vitamin C.
Vitamin C, good. So humans and other primates are the only mammals that cannot produce our own vitamin C. Every other mammal can produce its own vitamin C. We cannot. It gets even trickier because we have the gene to make vitamin C. It's just that there's a mutation, so our gene is broken, and all the primates, their gene is broken. So we all have the gene to make vitamin C, but because it's broken, it's a useless gene. It's mutated, it's useless, it's just stuck in our genome, and, there's no editing process, right? There's no natural selection reason for that gene to just disappear. It's just broken, so there's no reason for it to disappear. It's interesting stuff. There's a lot of really interesting examples, I think. Anyway, so a good question. Any other questions about the G1 checkpoint? All right, let's talk about the G2 checkpoint. Obviously, this is coming after G2. So obviously this is coming before the mitotic phase. So then logically, this first bullet point should make sense. The G2 checkpoint bars entry into the mitotic phase if the right conditions are not met. So once again, it's just going to stop the process if things aren't perfect, right? Or at least in theory. As we've just discussed on that last slide, it does not always work. But yeah, that's the purpose of the G2 checkpoint. It's almost like the last one before, well, it is the last one before finally getting into mitosis. The main purpose of the G2 checkpoint is to check the cell size and the protein reserves, which should make sense, right? When you think about it, it would make sense that in G1, you know what, actually, I'm not going to muddy the waters. We'll just leave it at that. Just leave it what's up here. And once again, with this checkpoint, we're going to ensure that the chromosomes have been duplicated properly and that they're not damaged and that there's not mutations. So there you go. 2 different checkpoints so far. Any questions about the G2 checkpoint?
What could cause the cell to be damaged?
Oof. Chemicals, like certain chemicals, UV rays, tobacco, alcohol, things like that. Yeah. So all that stuff they say is bad for you. Yeah. That's how it's bad. It basically, when you think of carcinogens, anything that could cause cancer, that's this. And we're going to get to that soon because this is where cancer comes from. When this stuff screws up, that's where we get cancer. And it's probably, unfortunately, one of the more relevant things you're going to learn in this semester. Because unfortunately, probably everybody in this room has either had cancer, will have cancer, or knows somebody who will have cancer. Especially as humans get to live longer. Because again, you right now, as you're sitting here, you're going through mitosis, right? You're making copies of your cells. The older you get, it's just a copy of a copy of a copy of a copy of a copy. So obviously, the older you get, the more chances there are of having this screw up. Anyway, any other questions about the G2 checkpoint? We'll talk about the last one, the M checkpoint. This is the one that's near the end of metaphase. It is also known as the spindle checkpoint. This is a good throwback. This is a good reminder. So this slide isn't very important for for the exam because I'm not gonna ask any questions about the checkpoints. The reason this slide is good is because it's reminding you of stuff you definitely need to know, which is that, remember, it's the spindle that attaches to the chromosomes and mitosis, and those are the things that move the chromosomes around, right? So in the M checkpoint, when all those things are lined up, when all those chromosomes are lined up, the cell makes sure that the chromatids are attached. And this also, when it screws up, is a big deal. We're going to talk about this too. Sometimes we get too many chromosomes because they didn't split properly. Sometimes we get not enough chromosomes because they didn't split properly. Very often that's deadly. And like for example, if you were to get pregnant or get somebody pregnant, and they had one of those mutations where there was too many chromosomes, a lot of times it just won't. How do I put it? Like a miscarry itself, I guess you could say. But then obviously sometimes people can survive it. And I'm sure you guys, and we're going to talk about this formally a little bit later, but a good example of that is Down syndrome. I'm sure you've heard of that. That's also called trisomy 21. So that is when an individual has an extra 21st chromosome. And obviously, I'm sure you've seen people with Down syndrome. They survive. It's not deadly, but there are obviously issues with it. And again, we'll formally talk about that later. Right now, that was just an example. The next word for attendance, I'm going to point to it. I'm not even going to say it. Is again, as a reminder, if you're online, you need to send those words before 9 a.m. to be considered present. The rest of you can just send them in for extra credit. Any questions about this slide? All right, so again, this is what happens in a perfect world, right? We have the G1 checkpoint. We have the G2 checkpoint. We have the M checkpoint. This prevents mutations. This prevents having too many or not enough chromosomes. But what happens when it messes up? And that's what we're going to talk about here. We're going to talk about cancer. Because basically what cancer is cells just dividing out of control. So like I said earlier, this whole process, the cell cycle, is controlled. There's on and off switches. There's proteins that say, all right, this cell needs to stop moving forward. It's not ready to go forward. Or there's proteins that say, all right, it's time to get moving with the cell cycle. But what happens with cancer is basically those proteins, those checks and balances, those start and stop signals stop working, and the cells just divide out of control. So, as your book says, cancer is a disease of the cell cycle. So like I already said, before I pulled this slide up, there's proteins that are in charge of the cell cycle. There's proteins that are the start and stop signals. So if you have mutations in those proteins, then obviously things are not going to work right, including stopping and starting the cell cycle. And because of that, it results in uncontrolled growth, and that is what cancer is. So, what are cancer cells? Well, they don't respond normally to the control system. Again, there's something wrong with the control system. There's something wrong with the start and stop signals. So again, it's just controlling uncontrolled growth. They divide excessively, and because of that, they also may invade other tissues, and that's where it gets deadly for us, right? Once it starts invading other tissues, and then whatever type of tissue that was, those stop doing their functions, and that's generally what kills you. Any questions so far? There's still more to talk about with cancer, but that's the introduction.
Some of these cell cycles take 20 minutes to do its thing. How quick is the cancer cell when it starts growing?
I think it would depend on the type of cancer.
Each cancer has a different cycle.
Yeah, I would think. And I honestly don't want to say that for sure. Be good independent work topic. Like, is there a difference between, brain cancer and liver cancer, for example? Or maybe the textbook says, and I don't remember it because I've only read that textbook once. But yeah, now I'm going to look that up myself. Any other questions? That's a great question. All right, some words you need to know. Proto-oncogenes and oncogenes, right? You need, actually, you know what? I'm going to put a big question mark next to this. I'm not quite sure I'm going to have this on the exam. It's obviously very important for your everyday life. This is very relevant, but sadly, or not sadly, but even though it's very important to your everyday life, I might not ask any questions about it on the exam. Anyway, proto-oncogenes. These are the genes that are responsible for the positive cell cycle regulators. So in other words, these are the ones that work, right? These are just normal genes This is the things that make the start and stop signals work. And then you have oncogenes, which are mutated versions of the proto-oncogenes, and they can cause cells to become cancerous. They're usually not harmful. So, for example, if there was a mutation that stops the cell cycle, that mutation is not going to get copied, right? Because if there's a mutation that won't allow that cell to move forward through the cell cycle, then it's not going to go to the S phase. Therefore, it's not going to duplicate its DNA. Therefore, it's not going to reproduce itself, and therefore that mutation is just lost, if you will, right? That's a dead cell, basically. One day, that cell's going to die, it's not going to reproduce, and that's the end of it. However, sometimes it is harmful. So if the mutation is such that it increases the cell cycle instead of stopping it, then obviously that mutation will be copied because that cell is going to go into the S phase, and that those chromosomes are going to be duplicated, including that mutation. So again, that's how it works, right? You get a mutation, the mutation's bad, and the mutation is copied. Every time you copy that cell, you copied that mutation, right? So you have one cell that has that mutation, and then it splits, so now you have 2 cells with that mutation. And then they split, and now you have 4 cells, and then 8 cells, and 16, 32, 64, right? So on and so forth. They keep doubling. But again, for the exam, this is probably not very important. If anything, maybe just know that proto-oncogenes are kind of like when things are working and everything's fine, but then the oncogenes are when things have gone wrong. Sometimes it's deadly when the mutation is copied. Sometimes it's not deadly because it just stops the cell cycle altogether. Any questions about that side?
I got a question a little bit different though.
Okay.
When the cell is mutation, whatever, it's dead, it's killed, does the body remove this completely out of your system? Or does the DNA stay in your
Oh, good. No, it gets basically digested, if you will.
digestive?
Yeah, basically, it's just the whole thing just gets disassembled and used for parts, including the DNA. So yeah, once this cell dies, for that bullet point, where it has that mutation that stops the cell cycle, it's that whole mutation it dies with it. It dies with the cell. Good question. Of course, this is a blanket statement too. There are other things like later in the semester you're going to learn about viruses, and there are some viruses that can even cause cancer. So again, everything I'm teaching you, there's always exceptions to the rule. And we're just scratching the surface, right? It's a 100 level class, not for biology majors. So as you might imagine, it gets a lot deeper than what we're learning here. But this is a good starting point. So again, sadly, if you ever have to do research about cancer, you're not going to be starting from scratch, right? You're going to have a good base knowledge. Any other questions about that before we talk about tumors a little bit? All right, tumor suppressor genes. It's in the name, right? You can tell what they do. They suppress tumors. These things code for the negative regulator proteins. In other words, the negative regulator proteins, remember I said they're stop and go signals, right? So the negative regulator control proteins are the stop signals. Let's put an S, right? S for stop. Maybe if I can try to draw an octagon. That looks horrible. Anyway, that's supposed to be a stop sign. Yeah, so the negative regulator proteins are the stop signals, and these are also tumor suppressor genes. These proteins prevent the uncontrolled division, which is what a tumor basically arises from, is that uncontrolled division. And a mutated suppressor gene results in genes that divide without the checking points. So all those 3 checking points that we talked about, we're just getting really specific here and saying if there's a mutation in the genes responsible for those checkpoints, then this is what we're talking about, right? Tumor suppressor genes. So all that stuff you learned about the G1 checkpoint, the G2 checkpoint, the metaphase checkpoint, that's this. So if something goes wrong, which is this 3rd bullet point right here, then that means there's no quality control, which obviously is not good. And then you're more likely to have mutations because there's nothing checking on it to make sure that things are the way they should be. Or again, like I said earlier, too many chromosomes or perhaps not enough chromosomes. So any questions about tumor suppressor genes?
What's QC stand for?
Quality control.
Quality.
No quality control. Or in other words, none of the G1 checkpoint, G2 checkpoint, or the metaphase checkpoint. And again, just as a reminder, as important as this is for your life, as far as the exam is concerned, it's not very important because I don't have probably any questions about it on the exam, but if so, there's not many. Here's a good example. So there's a P53 gene. You don't need to know this. This is just an example. I'm going to put a big X to it before we talk about it. I'm definitely not going to ask any questions about this on the exam. There might be questions on the quiz, but not the exam. So let's look at the difference between a normal P53 gene and a mutated P53 gene. In a normal P53 gene, it can detect this DNA damage, right? If there's DNA damage, it can detect it. It can detect cell cycle abnormalities, right? But if it's mutated, then that's not going to work. So basically, let me back up one step again, sorry. This thing we just talked about, these tumor suppressor genes, P53 is an example of them, right? So when P53 is doing its job, then it's making sure that the cell does not move forward if it has a problem, right? So sometimes it'll stop it and fix the problem. If the problem can't be fixed, like he's asked already, then we have this thing right here called apoptosis. So the cell recognizes and says, all right, this thing's damaged. It should not be copied because it has a mutation or whatever the problem may be. And it basically kills itself. That's when the thing is working. When you have a mutation in this gene, which is what's represented right there, there's no quality control. So you might have a mutation, but the gene, the P53 gene that's supposed to be checking on these things, well, the protein, it's supposed to be checking on these things, it's not working, right? Because you have a mutation. Therefore, even though you might have some damage or some mutations, whatever it is, the cell cycle is going to continue. It's not going to stop. And again, that is where we get cancer from. Because cancer is the disease of the cell cycle. And this is just one example of a protein that's involved in the start-stop signals, right? This is one of our quality control proteins. And when it is mutated and it's not doing its job, then there goes our quality control. Another thing, not that you need to know this, but I find this interesting. Humans do this to things on purpose. Like a lot of crops, like non-GMO crops. This is how they started. They were like they would take plants and they blast them with radiation and just see what kind of mutations they have. Sometimes they're good mutations and they keep them, and sometimes they're not. It's sort of like, well, it is. It's artificial selection, right? So sometimes humans induce these kind of mutations. Anyway, any questions about the P53 gene, which is just an example, and there'll be no questions about it on the exam. Okay. Well, shoot. 8:28, let's at least start the next one. Are there any questions while I pull up the next PowerPoint? I told myself this morning, I said I should get here early because I bet we're going to finish that chapter, and I bet I'm going to need a new PowerPoint. Didn't listen to myself, so here we are. Sorry about that. And it's being very slow. We wait. Oh, there we go. All right. Where are we at here? My courses. This is incredible stuff. Seven, 6, chapter 8. We're getting there. Chapter 7. That's it. Yes. Cellular basis of inheritance. What we're learning here is going to be basically genetics. Get it working. So I got some good questions about attendance words. Not to be a jerk, but if you miss the attendance words, you just miss the attendance word. That's the point, right? It's just to pay attention to get them. But keep in mind too, this is all extra credit. So for the person online saying that you didn't get one of the words, don't worry about it. That just means you're going to get a little bit less extra credit. You're still getting extra credit, just a little bit less, and you will still be considered present. So unless you miss all the attendance words, then you're good. Anyway, here we go. Finally, got it working. Chapter 7, the cellular basis of inheritance. So again, basically what we're about to learn about is inheritance, genetics. To me, this is probably one of the more interesting chapters, I think, because there's just so much stuff involved in this. Yeah, we'll get to it. Anyway, let's at least start. First of all, as you know from the last chapter, we learned about sexual reproduction, right? Sexual reproduction produces offspring that has a unique combination of genes from the parents. Actually, sorry, let me take a step back. I messed up. So yes, this is chapter 7. This is the cellular basis of inheritance, but I still need to get my brain used to this new textbook, because in the old textbook, mitosis and meiosis was one chapter, right? So remember, we just finished talking about mitosis, which is about asexual reproduction. And then again, with the old textbook, it was also meiosis, which is about sexual reproduction. But in this case, your textbook breaks it up into separate chapters. So here we go. Not only are we going to learn about genetics, but we're also going to learn about sexual reproduction and meiosis. So that being said, let's go back to where we started here. As you already know from the last chapter when we mentioned this, sexual reproduction results in the production of offspring that have a unique combination of genes from their parents. You already know this, right? You are not a perfect copy of your mom or your dad. You are a unique blend of your mom and your dad, the genes from your parents. And this whole thing depends on a process called meiosis. That's what you're about to learn about in meiosis. So again, we just got finished talking about mitosis, which is just regular old cell division or asexual reproduction. Sometimes, like right now, you guys are doing that. You guys are going through mitosis as you sit here. Some of your cells are reproducing. But now we're talking about sexual reproduction, and it's a different type of cell cycle. So let's talk about that. My recommendation, and you can do it however you want, but I recommend when you're studying this, make sure you understand mitosis before you start learning meiosis. Or let me rephrase that. Make sure you've memorized all the things you need to know for mitosis, and then start memorizing the things about meiosis. Because in a sense, the questions about meiosis are almost going to be like comparing and contrasting. So you need to know everything about mitosis, but then the questions about meiosis are going to be more like, all right, how is meiosis different than mitosis? And how is meiosis the same as mitosis? So keep that in mind, in the back of your mind as we're going through this. The questions for this portion of the exam are going to be very much comparing and contrasting asexual reproduction with sexual reproduction, aka mitosis with meiosis. And the first thing you need to understand is something about homologous chromosomes. So remember, before we talked about mitosis, we talked about sister chromatids, and I said that was an important term because one of the ways you determine the difference between mitosis and meiosis is by what the chromosomes do. So last time you learned about sister chromatids, and now I'm giving you a new word that you definitely need to know, homologous chromosomes. So let's talk about what those are. First of all, another thing you need to know is that different individuals of a species have the same number of chromosomes. So every genetically normal human, I'll put it that way, has the same number of chromosomes, right? So unless there's something abnormal about any of you in here or myself, you all have 23 pairs of chromosomes because you are a human. And part of being human is the fact that you have 23 pairs of chromosomes, aka 46 chromosomes. So those are numbers you definitely need to know. You need to know that we have 46, the humans have 46 chromosomes. Oops. Excuse me. AKA 23 pairs of chromosomes. Another term you need to know is this one, somatic cell. I might ask about that on the exam, but I'm definitely going to use the term. So you need to know what it means when I use the term. A somatic cell is basically anything that's not your sperm or egg. So any cell in your body that's not a sperm or egg, essentially is called a somatic cell. Any questions about that?
So somatic cells is your like lung tissues stuff like that.
Yep, brain cells, kidney
Brain
cells,
cells.
lung cells, eyeball cells.
So what do they call the sperm and the egg cell?
We'll get to that. Good question.
Okay.
Great question. Well, I'll go ahead and tell you, but I'll tell you again later, but it's called gametes. Gametes, yep. Anyway, another thing I might ask you about this, you're definitely gonna use this on the lab. Is that this week's lab? Yes, this week's lab too. You're gonna use something called a karyotype, right? And a karyotype is basically just a picture of all your chromosomes. So let's talk about that really quickly. To produce a karyotype, a scientist would have to break open a human cell, or any cell it's gonna look at, but we're just talking to humans, because why not? And they do that during metaphase of mitosis. If they want to look at the chromosomes, why do you think they'd go for metaphase? Why not anaphase or telophase or prophase or interphase for that matter? Why metaphase? Yeah, they're all lined up, right? They're all condensed because they're in mitosis, and they're all lined up in the middle of the cell, so it's nice and easy, easier to see them. Yeah, so that's why they're done in during mitosis. I'm going to put it next to that though, because I'm not going to ask you that question in the exam. So after they've opened up the cell in the middle of metaphase, then they also have to stain the chromosomes so that they can be visible to the naked eye. I'm also not going to ask you that. Then obviously they're going to take a picture with a microscope because you can't see your chromosomes with your naked eyes. So there you go. I'm not going to put it next to that because I don't know, I might ask you that, but it's kind of a given, right? You're going to need to take a picture of it. You can't just use your cell phone. So there you go. Take a picture with a microscope. And then they kind of like cut and paste it, right? They can copy it and move them around, and they arrange the chromosomes in matching pairs by size. So it's always the largest at first, and they get smaller and smaller and smaller until you get to the smallest chromosomes at the end. So that's how a karyotype works. And again, you're going to use one or a few of them for this week's lab. So this is a normal karyotype. We can see here 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30, 32, 34, 36, 38, 40, 42. And I'm about to talk about those at the bottom, but yeah, you can see that each one of those chromosomes they're in a pair, right? So there's 2 of the same chromosomes, 2 of the same chromosomes, or homologous chromosomes, 2 homologous chromosomes, 2 homologous chromosomes. So if there was something wrong with your chromosome number, this is what you can see with the karyotype. And that's what you're going to be doing in one of the labs. You're going to basically be pretending like you're a doctor, and I'm going to give you a picture of a karyotype, and you need to choose. So you're going to see like, oh, there's an extra chromosome on that one, or there's a missing chromosome on that one. And there's a little table, and you get to use that table to diagnose the problem. But again, you can see here, this is the normal karyotype. There's 2 of each chromosome. And sometimes there will be doubles. Sometimes there's not going to be enough. And also, just so you know, and you're going to get really familiar with this in the lab, they are also numbered. So it starts at 1 and ends at 23. And actually, we don't even use the number 23. You'll see why here in a second. So there's another picture of a karyotype. This one's colored. It's easier to see. But again, 23 pairs of chromosomes. But again, like I said, the last pair, we don't go by the number. They're either XX or they're XY. We'll talk about that in more detail too. But for now, that's a good introduction. So any questions about this karyotype? Excuse me. All right. So let's talk about these homologous chromosomes. You have a pair of each of your chromosomes, right? One of them you got from your mom, one of them you got from your dad. So for every chromosome in your body, unless something went wrong, you should have 2 pairs of them, 1 pair of each chromosome, and one of them came from your mom, one of them came from your dad. And again, they're the same chromosome. So I'm going to randomly say chromosome number 3. You got 1 chromosome number 3 from your mom, you got 1 chromosome number 3 from your dad. They're the same size. They have the same centromere position. And I guess I'll try to draw this to remind you of what that is. Really bad at drawing, especially when I have to use a mouse to draw with it. Like if that's a chromosome, and that right there is the centromere, right? Sometimes it's in the middle. Sometimes you might find it more up towards the top. Sometimes you might find it more towards the other end. But anyway, a homologous chromosome, again, it's the same size, has the same centromere position, which can vary. And more importantly for you guys, in this part, I put it in bold because you definitely need to know this. They carry the same genes. So again, randomly, I'm just making something up. I don't know this, but I don't know. If there was a gene for a hairy back on chromosome number 17, that means you got one version of that gene from your mom. It's on the same chromosome number 17 at the same place as the one from your dad. And not only that, but it would be the same for all of us. So you can look at the hairy back gene that I'm making up on chromosome number 17. Let's say it's right here at the tip of chromosome number 17. If you were to look at my DNA, I would have the gene for the hairy back gene at the tip of my chromosome 17. And you all would. You would have the same gene in the same place because we are all humans, and that's how it works, right? The chromosomes, again, homologous chromosomes are the same thing, it's just different versions of them. So you got your mom version and your dad version. They carry the same genes at the same spot, but there are different versions of the genes, and we'll talk about that later in this chapter too. So these genes also control the same characteristics. It's very important. This is a very, very important bullet point right here. Make sure you're very familiar with this bullet point. Because not only is this going to be important for understanding meiosis, which is what we're getting at here, we're trying to get to talking about meiosis, but when you start learning about how genes work, you really need to understand how homologous chromosomes work. So once again, the second bullet point, I've already said this, but here it is again in writing. Humans have 46 chromosomes, which means we have 23 pairs of chromosomes, right? So they're all numbered 1 through 22, and then of course 23 is your sex chromosomes, and we'll talk about that later. Actually, we'll talk about that right now, because there it is. For humans, a male is an XY. For females, the chromosome is XX. This is a 100 level biology class, so this is the basic definition of a male and a female. You're going to learn in this class there are some exceptions to this. Sometimes there's extra chromosomes, sex chromosomes. Sometimes there's missing sex chromosomes. And of course, we don't even dig into anything deeper than that in this class, but then there's also things like, some people are born with all parts, right? So some people have a vagina and a penis, and their chromosomes reflect that. So what we're teaching, again, is the basic version of a very complicated, I don't want to say situation, but complicated study, if you will. Anyway, again, for the exam, you definitely need to know, very important slide, everything on here you need to know. You need to know what homologous chromosomes are. You need to know that humans have 22 pairs of autosomes. Oh, yeah, sorry, that's a word I need to teach you too. Autosomes are any chromosomes that aren't sex chromosomes are called autosomes. We have 22 pairs of them, and then we also have one pair of sex chromosomes. And males are XY, females are XX. You need to know all of this for the exam. As far as the exam is concerned, this is probably the most information-dense slide that I've shown you. You need to know all that. So any questions about that? All right, next word for attendance will be parent. I circled it right there, parent. Any questions about the slide? We're good, okay. Let's move forward. So, like Kesterson already asked, remember I told you that all your cells are called somatic cells, other than your sex cells, which are your egg or your sperm. And then he was asking, well, what do you call the sex cells? And that's what I'm about to tell you. They are called gametes. So let's talk about gametes and the life cycle of a sexual organism. Actually, you know what? Let's see, it's 8:44. Let's just send it a little early, right? So when we get back, then we'll talk about gametes and the cell cycle. So that's it for today. Remember, my office hours are going to be online only today. So if you need me, leave me online.